Hey guys, today I'm doing a presentation on post-translational modification of human histones by wide tolerance of acetylation. This experiment was done by extracting human histone proteins isolated by kidney cells adjacent to kidney cancer in 12 patients who have undergone nephrectomies. The histones H1, H2, A, B, H3, and H4 were separately excised by HDS Page gel and digested in gel with trypsin, then extracted and desalted peptides were subject to peptide fractionation by liquid chromatography and then mass spectrometry to look for a mass of 42 plus or minus 0.5 Daltons, which corresponds to modifications of acetylation and trimethylation. If you look here at figure 1a, it shows the MS spectra, which were searched against the human histone database using Mascot and Sequest, a total of 158, 88%, 108, 97%, 54, 96%, and 22, 90% peptides were identified for human histone H1, H2, A, B, H3, and H4 respectively. Here shown are the histones excised, and then in figure 1b, after the MS spectra of histones were searched using 42 plus or minus 0.5 Dalton delta mass, the frequency of modified peptides was plotted and shown. 213 modified histone peptides were found with 42 plus or minus 0.5 Dalton modifications at lysine, alanine, threonine, serine, and glutamic acid, but most frequently at lysine, serine, and threonine. Okay, they were looking for two modifications, acetylation and trimethylation. And then they realized that amino acid substitutions also fell within their range of 42 plus or minus 0.5 Daltons, shown here in figure 2a. The serine to glutamic acid, acetylation, adenine to arginine, and trimethylation. In figure 2b, to confirm modifications and get rid of the data as shown above, the acquired MS data was further searched with more sophisticated algorithm using Sequest. A total of 138 spectra of modified residues were identified. Among 79 or 57% were acetylation and 59 or 43% were trimethylation. The distribution of acetylated and trimethylated residues were found mainly on H3, as shown in figure 2a or 2b. And then figure 3 shows that they had found new acetylation sites that could be areas of further research into the regulation of histone structure and function. Then figure 4a and 4b are validation of the T22 in human histone H3. Figure A shows that the anti-H3T22AC antibody selectively recognized a peptide with the acetylated residue H3T22AC, but did not detect the same peptide without the acetylated residue, or different peptides with acetylated serine at a different site. Figure 4b shows that the anti-H3T-AC antibody was subsequently used to detect the acetylated H3T22AC in HEK293T and HELA cell lysates by the Western blot assay, shown here.